Today we're going to be looking at topic 2, cells and their organisation. This is topic 2 in the Edexcel Biology, IGCSE Biology. First syllabus statement, 2.1. Describe the levels of organisation in organisms, organelles, cells, tissues, organs and systems. Starting from smallest to biggest, organelles are smallest, then the cells, then tissues, then organs, then the organ system, then the organism itself. Here are some definitions of each level of organisation. These are very common 1-mark answers. You should be able to obtain the easy mark. Organelles. Cell structures specialised with a specific function. For example, a nucleus. Cells. Basic functional and structural units in a living organism. An example would be a nerve cell. Tissues. Composed of same cells carrying out the same function. Organs. Comprised of different tissues carrying out a particular function. An example would be the heart. An organ system. Composed of a group of organs working together to perform a body function. For example, the digestive system. That is the first syllabus statement of 2.1. 2.2 and 2.3. I've put these together because they make more sense and a lot of crossovers between them. Describe cell structures including the nucleus, cytoplasm, cell membrane, cell wall, mitochondria, chloroplast, ribosomes and vacuole. Plus, describe the functions of all of those organelles. So it's a big syllabus statement, but it's really not that hard. Here we go. A nucleus. The nucleus contains genetic material in the form of DNA, which forms chromosomes. The function is that it controls the activity of a cell. The cytoplasm. The cytoplasm structure is a gel-like substance. This is where most of the chemical reactions happen. In order to control these reactions, there are many enzymes which catalyze the metabolic reactions. The organelles are located within the cytoplasm. The cell membrane. This controls what goes in and out of the cell, often with selectively permeable membranes. It also holds the cell together. Cell wall. The cell wall provides structure to the cell and strengthens the cell itself. It is very strong and thus keeps a rigid shape and structure. Mitochondria. Mitochondria is sausage-shaped. This is where the reactions for aerobic respiration take place. The aerobic respiration transfers energy in the form of ATP that cells need to function. Ribosomes. Ribosomes are very small particles. This is a site of protein synthesis. Protein synthesis is essentially just a production of proteins. Vacuole. The vacuole is a sac within plants that maintains the internal pressure to support and provide structure for the cell. It contains cell sap consisting of sugar and salts. Chloroplast. Chloroplasts are disc-shaped. This is where photosynthesis occurs. It contains a green pigment called chlorophyll that absorbs light energy, which later becomes converted into chemical energy. So those are all the organelles that you need to know in this syllabus statement. Here are some diagrams of a plant cell and an animal cell. It is important to be able to label both of these diagrams. As you can see, there are more organelles in the plant cell than an animal cell. 2.4. Know the similarities and differences in the structure of a plant and animal cell. The similarities are, they both contain a nucleus. As you can see here, they both have a nucleus. They both have ribosomes. You can't see ribosomes here, but it's important to label them on your diagram in your exam. Ribosomes are very, very small and microscopic and is where protein synthesis happens, as mentioned before. They both contain mitochondria. As you can see here, the pink. They both contain a cytoplasm. This is the gel-like substance in which most of the organelles are in. Differences between them. A plant cell contains chloroplasts. As you can see here, these green things. This is what enables the plant to do photosynthesis. A plant cell contains a vacuole. As you can see here, this larger thing. Some animal cells contain a vacuole, but it's often very small and often temporary. Plant cells. Plant cells contain a cell wall. However, an animal cell only contains a cell membrane. This is an important difference to note. 2.5. Explain the importance of cell differentiation in the development of specialized cells. This is only a triple award only. So if you're not doing triple award, you do not need to know this. The important definitions here. These are very common 1-mark questions, so be confident. Cell differentiation. The process by which newly formed cells become specialized to carry out a specific function. Definition number 2 of a specialized cell. A type of cell that performs a specific function soon after the process of cell differentiation. Here is a step-by-step process of cell differentiation. Step 1. An undifferentiated cell receives a signal, which therefore stimulates a change of shape in the cell, as well as metabolism. This therefore enables the cell to carry out a specific function and become a specialized cell as such. This therefore means that the cell can now develop cells required to develop and meet growth requirements. For example, embryonic stem cells could differentiate into a nerve cell. Here are some other examples of specialized cell: sperm cells, ciliated cells, and egg cells. 2.6. Understand the advantages and disadvantages of using stem cells in medicine. This is another triple award only. This is part of the new spec, so you probably won't find this topic in past papers, so be sure not to forget about it. The definition of a stem cell: a cell that is said to be unspecialized and possesses the ability to become specialized to perform a specific function. Stem cells can be found in embryos, adult tissue, in the bone marrow, and umbilical cord/placenta. The advantages: firstly, they are unspecialized and have the ability to become specialized, allowing for medical benefits. Number 2, potential of discovering new treatments to existing conditions. Number 3, ability to test stem cell drugs without the use of mammal simulation. Number 4, there can be a reduced risk of rejection if patients' own cells can be obtained and used. Number 4, ethical issues. Some see it as the destruction of potential life. Number 2, people are unsure about the long-term effects of stem cell therapy. Number 3, there may be a risk of rejection by the patient's own body. And number 4, it can be highly difficult to make the cells become specialized. Here's a diagram showing what stem cells can become into. Do not ever refer to the mitochondria as the powerhouse of the cell, as this is key stage 3, and furthermore, do not refer to the nucleus as the brain of the cell. So this is that topic, and you can find all the notes on the website link below.